Hello, everyone. Welcome and thank you for joining us today on another episode for Emerge. My name is Stoyan Stoichev. I'm part of the Resin Bio team. Today we have a little bit of a different session. We're going to try for the first time do a hands-on tutorial. And for this, I have the pleasure of hosting Devon Kohler from August Wittes lab. He's doing his PhD there. And his focus is, as you expect from August lab, is on statistical analysis, specifically on proteomics datasets. And Devon has been really instrumental in developing some of the tools around MSStats and R packages, developing the MSStats PTM, MSStats Shiny, and so on. And so today he will take us through some of these packages, how we can run them, and some of the implication of the different variables you can set in terms of the data outputs and how the data will look at the back end. So I think because it's a hands-on session, it's good that you can post your questions as we go along, and we're trying to tackle these as the session goes. We don't have really kind of a time limit, so we can take it easy. But yeah, if there's very technical questions in terms of installation and so on, maybe we'll tackle those offline so we don't slow the session too much, but still post them in the Q&A section, and we'll try and answer them at some point. So Devon, thank you very much for joining us today. It's really a pleasure to host you and to have this first hands-on session on Emerge.
Awesome. Thanks, Stoyan. Okay. Welcome, everyone. So as Stoyan mentioned, I'll be going over the methods in MSStats and MSStats TMT, and also showing a couple case studies on how to apply the methods. So I think Stoyan touched on this, but real quick, some background on me. I'm a PhD student in Olga Witek's lab at Northeastern University. Before my PhD, I got my master's in data science from Northeastern. My main research areas are in statistical and causal inference in MS-based proteomics. And I'm the lead developer of MSStats and the creator of a couple different MSStats packages. So before we begin, I just wanted to go over kind of the schedule for the webinar today. First, we'll start with an overview of MSStats, what MSStats is, what the objectives of the package are, and kind of where it fits into the proteomic analysis workflow. Then I will go through some methods specifically as they relate to label-free DIA, although they can be expanded to other acquisition types like DDA, SRM. We'll talk about the implementation in MSStats Shiny. Then we'll go into the hands-on session for DIA specifically. We'll come back to the methods section and do some expansions to TMT because there are some different methods there. And then we'll do the hands-on session for MSStats TMT. Okay, so first off, what is MSStats? Originally, MSStats was a single R bioconductor package, but more recently it's expanded into a number of different packages. So you might hear someone say MSStats PTM, MSStats TMT. It's all a family of packages which we call MSStats. The overall goal of MSStats, regardless of the package you use, is to detect which proteins change in abundance between conditions. And the packages can handle complex designs, like factorial experiments, paired design, time courses, anything like that. And they also provide some auxiliary functions and outputs, such as the protein abundance of each subject on a relative scale, which can be used for different applications other than differential analysis, such as maybe machine learning methods like PCA, which are really helpful to have the intensities on a relative scale. Now, we can handle a whole bunch of different types of experimental designs through these different packages. They can be label-free or label-based. So it can be DDA label-free or DDA with TMT, DIA, PRM, SRM. And we can also handle different biological questions. So we can detect differential post-translational modifications, And we also have a package for limited proteolysis, where we're trying to detect changes in protein structure. Regardless of the experimental design, MSStats will always take the same type of input. So MSStats takes as input the output of different spectral processing tools like Skyline, MaxQuant, FragPipe, Diane, anything like that. MSStats won't do any identification or quantification of peaks. We rely on other tools to do that, but then we take the output of those tools as input to our models. And we have specific converters for all of these tools so that you can convert them directly into the format that we need and make it easier for the user. And there's different data formatting methods, cleaning, QC that are involved in all of the converters. All of these tools output things and make different decisions in different formats. And we'll talk about that in a bit, but we want to be able to take the output of any of these tools and use them for MSStats. Now, once you're in the MSStats workflow, we have a variety of statistical methods that are flexible and can account for different complex designs, different missing values, outliers, and they estimate all of the parameters in a stable way so that when you apply the statistical methods, you're not just getting some weird values that don't make any sense. Now, while we have all of these complex methods, this is all behind the scenes. So the user doesn't have to specify their experimental design or their specific model in the code. We'll detect that for you from your input, which is really nice because it takes a lot of the statistical burden off of the user, and you don't need to worry about what model is correct for your data. After statistical analysis, something that we've been doing and we'd like to do more is trying to treat your experiment as like a pilot study for future experiments. So this is really talking about method validation, system suitability, and experimental design. And what we can do is take this experiment that you processed before, look at the variance in the experiment, and use that variance to try to determine how many replicates you would need to reach a certain statistical power in future experiments, and help you determine how many samples you'll need going forward for an experiment that you like the one you just ran. And I wanted to point out that all of these methods are published in a recent JPR paper on MSStats version 4.0. And if you want more in-depth descriptions of the methods in MSStats, you can go to that paper. It's the best resource currently for MSStats. In particular, I'd like to point you to the supplementary because there's different things with annotation files, which are really important, which we really go into depth there. And I'll talk about annotation files in a little bit, but I just wanted to point that out. Okay, so like I mentioned, MSStats is not a single package. It's an ecosystem or a family of packages, and this slide kind of highlights where they all fit in. But first, I wanted to highlight some of the key contributors to MSStats. First, Mina Choi, who is really the original and main developer of the MSStats package itself. She really put all the infrastructure around MSStats and was the original coder. She's now at Genentech. Melanie, who's done a lot of work on the Galaxy project, implementing MSStats into a GUI in the Galaxy interface. Ting, who's done a lot of work on MSStats TMT. And Mateus, who is now the lead developer of specifically the MSStats package, and he's done a lot of work refactoring the package to make it scalable and fast in with increasingly large data sets. And as we mentioned, I've done some work on everything, but specifically PTM, LIP, and MSStats Shiny. So up at the top, you can see the tools that we currently have converters for, and these are going to be dependent a little bit on the experimental design, but no matter the tool, we use MSStats convert to actually take the output and put it into the correct format. Now, MSStats convert isn't something you'll never have to use. It's kind of an underlying package to our other packages, but it's helpful if you do want to use it, if you want to maybe build a converter that we don't currently have for your workflow. Maybe I think we don't have like metamorphosis. So if you wanted to do something like that, you could use the MSStats convert functions. MSStats itself is specifically for label-free analysis of DDA, DIA, and SRM data, but it is used as the fundamental methods for our other packages. So all the packages call MSStats itself. MSStats TMT is, of course, for TMT experiments. PTM for post-translational modifications. And then we have these GUI tools, which we'll talk about MSStats Shiny, which take the code and put them in a point and click interface. So that's MSStats. And when I talk about MSStats, we either the specific package or it could be referring to the family of packages here. Okay, so where does MSStats fit into the workflow of your typical proteomics experiment? A lot of times people will focus a lot of their effort onto the data acquisition and quality control step. And while it's super important, from our point of view, there's actually a lot of work that has to go in to the experimental design before, and also a lot of steps when MSStats comes in data processing and analysis. And what I want to talk about specifically first is experimental design, because experimental design is really important. And if your experiment is not designed correctly, then no statistical analysis can actually solve those problems. And what I mean about experimental design is a couple things. First, we have to think about our study goals. So are we doing an exploratory analysis where we're just looking for interesting proteins? Are we doing more of a differential analysis where we're trying to detect differences between two populations? Or are we doing biomarker discovery where we're really interested in a single individual? Now, MSStats itself is really suited for differential analysis and not something like biomarker discovery. And why that is because differential analysis compares population means, whereas biomarker discovery will focus on the individual. So if you want to do differential analysis, that's where MSStats comes in. Beyond the study goals, there are both biological and technical aspects of the experiment. For biological aspects, it's different selections of your conditions. How many conditions do you have? Are they at different time periods? And then replicates, how many biological replicates or observations do we need in order to reach our desired statistical power? In terms of technical aspects, we have things like sample preparation, data acquisition, DDA, DIA, and randomization. And randomization is actually really important, especially when it comes into things like TMT, where we're going to have different mixtures with different channels. And we want to make sure that we have the correct randomization of conditions in our channels per mixture so that we're not biasing our experiment. And for an example of an experimental design, you can see here kind of an older experiment with 2 breast cancer cell lines. These were treated with different levels of oxygen and measured at different time points, different durations, multiple cultures per time point, and there were actually different technical replicates per culture. And what we want to do as MSStats is be able to detect all of these different sources of variation, determine what is a technical aspect, what's a technical variation, what's the true biological aspect, what's the 2 changes in different conditions, and be able to detect what proteins are differential between them all. So you can see the experiments can get very complex, so we need to be able to break everything down correctly. Another example experiment here, this is a hybrid design, a time series experiment, and you'll see that there were 29 healthy subjects, 14 disease subjects, and the disease subjects were measured at different time points. So we have measures before treatment, a bunch of different measurements after treatment at different time points, and some measures after surgery. And you can tell there are a whole bunch of different questions that we can ask here. You can compare healthy versus disease before treatment, healthy versus after surgery, maybe like an average of the different disease cases versus healthy. And because these are repeated measurements with the same biological replicates, the statistical methods need to be able to account for that and are required to be more complex. Okay. So after you've designed your experiment, you've acquired your data, done your quality control, this is where MSStats will come in. And as I mentioned before, MSStats takes the list of identified and quantified peaks from different spectral processing tools. Now, we try to be equal opportunity, so we don't just try to fit to one specific experiment or one specific tool. We don't want to just have our methods work for MaxQuant. We want them to work for any of these tools. And this is challenging because we want our biological conclusions to be independent of the tools as possible and have between tool reproducibility. And that essentially means no matter what tool you use, you'll get the same results from MS Stats. And that's a challenge because these tools have different steps. They have different ways of doing QC, different ways of normalizing, different ways of identifying missing values or outlying peaks, and different ways of quantifying proteins in a specific run. So what our converters will do is try to account for all of these differences and provide the same results no matter what tool you use. And to show you an idea of like how different these tools can output data, here you'll see two experiments, one DDA experiment, one DIA. These are histogram of feature intensities produced by different tools. So we have Skyline, MaxQuant, and Progenesis for the DDA, and Skyline and Spectronaut for the DIA. Now, first you'll notice just the different scales or the scales for each different tool are different themselves. So for Skyline and MaxQuant, the scales are right around 25, but for Progenesis, they're actually lower. They're around 20. And for the DIA, Spectronaut is significantly lower than Skyline. And now this isn't necessarily a problem for our methods because we look at the comparisons on a relative scale. But what is important is the number of peaks. So you can see like the number of peaks quantified are going to be different for Skyline, MaxQuant, and Progenesis. Progenesis actually has a lot less. And also in how these tools report missing values. So Skyline, for instance, you'll see it has a whole bunch of zeros, 2,400 zeros, and only a couple NAs, 219, whereas MaxQuant has no zeros reported, but a ton of NAs, about 40,000. And to give you a little insight of why this specific case is because Skyline only uses NA when the peak is present, but can't be quantified, whereas MaxQuant is much more liberal with their use of NAs. But regardless, we need to be able to know why these tools are putting in zeros or NAs and then be able to output the correct reason and do our converter so that the downstream statistical methods will be able to handle that. The last thing I want to talk about here is censoring because it comes up in our methods in a little bit. So you'll see in these histograms, we have these kind of larger striped red lines on the left-hand side of the histograms on the far left tail. So what MSStats does is we treat very low intensities as unreliable. So if they're very low, we think that they're pretty close to the limit of detection where the machine cannot detect these intensities. And so we don't really trust them. And basically what we do is we find this very low tail of the probability distribution. It's like the 99.9th percentile, and anything below that we'll treat as censored. And I just wanted to bring that up because we're going to be talking, you'll see some censored values later. Just so you know what censored is, we have missing values, which are NAs, and then censored values, which we actually censor ourselves. Now, regardless of the tool that you use, the output of the converters will always look the same. And this is the kind of classic MSStats. We call it the MSStats input, but it's actually the output of the converters and what's input into the rest of the pipeline. And just off the bat here, you might hear me say features a lot. And I just wanted to kind of go over what I mean by a feature. We define a feature dependent on your acquisition type. So it's going to be a precursor ion in DDA, a transition in SRM, or a fragment in DIA. And just so you know what I mean by features, because I say it all the time when I'm talking about these experiments. You'll see that this input data, though, is really standardized. So we have our protein name, the peptide sequence, a bunch of different columns that can indicate our feature type, the condition, bioreplicate, the run, and the untransformed intensity. And this is always what the output of these features of our converters will show. Okay. After getting the data into the correct format, we've dealt with the different selections by the converters. The next step is data processing. In particular here, I'm going to mention normalization because normalization is very important. So here you can see an example box plot for one experiment where we have our runs on the x-axis and the log intensity of all the features in the run on the y-axis. And you can see in this experiment, there's actually a pretty big difference in these box plots as determined by the run. So maybe in runs 1 through 5, maybe they're kind of lower, a little bit wider of a distribution, and then runs 6 through 7, it's a little bit tighter and a higher intensity on average. And the question is this true biological variation between the runs, or is this due to technical reason, just maybe there was some machine drift or something like that as you did your runs? And we account for this by normalizing. So for this specific experiment, we actually had some reference standards that were spiked in at a constant intensity across all runs. So we actually can tell here how much is due to biological variation and how much is due to technical variation. And what we'll end up doing is normalizing all of the spiked in reference standards. And then whatever we had to shift those reference standards by, we'll shift these, the rest of the proteins in the sample. So you can see that after we've normalized them, all of their quantiles and medians are equal, but the rest of the proteins actually didn't have to be shifted as much. So there is still some true biological variation as the runs go on. Now, I do want to mention, if we do something like these reference standards, we only are accounting for technical variation in terms of the machine's measurements. We're not going to be able to normalize for anything further upstream, such as digestion, sample storage, or other upstream steps. So those are all going to those won't be accounted for. Also, what do we do if we do not have reference standards? How do we normalize? And this is a challenging question. Generally with MSStats, our default is to equalize the medians across the proteins. So that would mean here how all of these medians are equalized. We would do that to this, all of the proteins in the sample. So all of these medians here would just be equalized across all of the runs. This is a good idea if you don't expect the majority of the proteins in your experiment to change. If you expect the majority of your proteins across the runs to be the same and only a couple proteins or a small amount of the proteins to change, this can work really well. And that's why it's the default. However, if you think that your experiment does have a lot of changes in proteins across the different runs, across the different conditions, then equalizing your medians is going to remove true biological variation from your experiment, and you will get less differential results. It will be missing differential proteins.
Devin, we have one question from Erin coming in that asked about the different normalization methods in terms of particularly quantile versus equalizing medians.
Let's see.
So the difference between these two.
Yeah. So the main difference is you can kind of see here the actual quantiles here are normalized. So all of the quantiles are exactly equal in these reference standards. When we talk about median normalization, we're going to be equalizing just that median value. So you can see like the quantiles would actually be different in median normalization, whereas here, these distributions are like exactly the same. And so equalizing the quantiles is a little bit more, I would say, intense, or it fits it a little bit more, where we're actually going to be normalizing things more, shifting them more so that they're all equal, compared to median normalization, which is just going to put that median together. So it's really going to depend on your specific experiment. And that's kind of something about this normalization that is really specific to, or that is really important, is that all of these are going to be specific to your experiment. So it's kind of hard to tell you right at the beginning what's going to be correct for your experiment. I actually bring up one issue we see sometimes is we do a lot of spike-in experiments where we can see like the true changes in the proteins. We know like they're spiked in at different ratios, and we know that there's a differential abundance. Sometimes if we do equalizing the medians, we'll actually remove that true variation that we expect to see. So that's a challenge that sometimes we run into as well. So I don't have a great answer for like something for all of the experiments. It's going to be specific to your experiment. Okay. And really beyond normalization, upstream processing in general is really important and can have a really big impact on the statistical model. So a lot of times what we'll see is people will reprocess the same data sets using a different method. And rather than the statistical models that have a major impact on the results, it's how they did their upstream processing that has the biggest results on the downstream differential analysis. And this is something that personally I'm really interested in, is trying to quantify the uncertainty in these upstream processes, just because of how important it is to our statistical methods. Okay, so after our data processing, now we can do differential analysis. And when we talk about differential analysis, what I really am talking about is fold changes and p-values. So here you can see an example volcano plot where on the x-axis we have our log fold change, and on the y-axis we have log adjusted p-values. And you can have different criteria for what you want to treat as a differential protein. Maybe you want the fold change to be a specific value or above a specific value. Maybe you want your p-values to be above or below a specific value, 0.05 or 0.01. But regardless of the case, this is how we will actually show that in the volcano plot, where we have upregulated and downregulated proteins. Other than volcano plots, another popular plot that we have in MS Stats is the heat map. And for the heat map, it's really nice if you have multiple comparisons. I think in particular, like time series comparisons where you're having different time points, maybe they go higher as you go along, and you can see just how your proteins will change in terms of their differential results, their fold change as those comparisons go along. The last step, kind of mentioned it before, but it's experimental design and trying to determine the correct number of sample size of replicates for future experiments. So we can treat your experiment as a pilot study. We'll look at the average variation in your proteins, and then determining, or depending on how much power you want in a future experiment, we can try to detect how many protein or how many biological replicates you should have going forward. One thing I want to mention here is that it's not just determined by the variance and how many or your power. It's also determined on your experimental design. So here you'll see the number of biological replicates here for a experiment to reach a certain power, and the desired fold change here. And these different lines correspond to different false discovery rates. So this top line corresponds to an experiment that is more of a discovery-based experiment where you don't expect many proteins to change. And this middle line is an experiment that's more targeted, and you expect to see a lot of changes in the proteins between conditions. When we have less proteins that we expect to be differential, we need more replicates to protect against false positives, because there's some probability that we're just going to have random proteins that aren't truly differential that we show as positive incorrectly. If we expect more proteins to become differential between groups, we don't need quite as many replicates. And this is also going to be dependent on your tolerance to have false positives, or your tolerance to have incorrect results. So if you can tolerate maybe more false positives, you can reduce the replicates that you use in your experiment. But if you have to be really stringent and make sure you're only seeing the true positives, then we need more. And this is just a long way of saying we can't really answer the question of how many replicates do I need off the bat. It's going to really depend on your experiment and the variance that you expect to see and your tolerance for error. Okay. So that was an overview of MS Stats, where it fits into the pipeline and the different steps of MS Stats. Now I'm going to go through some of the specific methods for label-free and specifically for DIA, but they do work for DDA, SRM, everything like that. And they're also really the fundamental methods we use for all of the packages. So these methods aren't just for MS Stats, they're used in PTM, TMT. They're just expanded upon in those packages. Okay, so this is how we look at an experiment for one protein in a label-free design. And we can see the different sources of variation here. So we have a different number of conditions. Per condition, we have a certain number of subjects or biological replicates. And then per biological replicate, we have different numbers of technical replicates. We also have different features which actually make up the protein intensities of the specific runs. So we have different features, which are technological variation. These are our biological variation. And then we also have, of course, like our censored values, which we talked about earlier, and our missing values for each run. And so what we want to do in MSStats is be able to represent this experimental design, represent the peak intensities, and really overall be able to estimate the different variance components, the variance of the features, the variance of the runs, the subjects, the conditions, and be able to do your inference on these conditions. So be able to detect, is this protein differential between condition 1 and condition 2? How we do that is we use what's called a split plot approach. And this is a well-studied method in statistics in general. And essentially what we do is we split this data table up into 2 specific parts, and then we deal with each part separately. So one part is called the whole plot, and that's where we do our condition, our subject, and our runs. And then we also have our subplot where we're only looking at the runs and the features. And this is the whole statistical model if we were to fit like the full model with the features. But what we found is that splitting it into 2 separate sections and working on the subplot first and then doing the whole plot modeling performs much better. And there's 2 specific things we do on the subplot. So we've talked, or I guess 3, we talked about normalization, which is the first step. After normalization, we do missing value imputation, and then we do summarization. So first I'm going to talk about missing value imputation. MSStats will treat missing values as missing due to reasons of low abundance. And you can see the statistical models here, and I'm not going to go too much into the statistical models, but what I can do is give you sort of some heuristics or intuition behind how we do missing value imputation. So this is an example profile plot for one protein with 3 features and 2 runs. The log feature intensity is on the y-axis and the runs are on the x-axis. You can see this is our original measurements without imputation. We have all of our measurements for MS run 2, but in the MS run 1, we're missing the third measurement for this third feature, or the measurement for this third feature. And MSStats is going to assume that it dropped below this limit of quantification and that's why it wasn't measured. Without imputation, we actually will overestimate how large the feature intensity was for this run. So you can see it's kind of high, a little bit higher than it should be. For more naive methods of imputation, if we're going to maybe impute with 0 or some very low value, we're going to overestimate the regulation or overestimate how low the value truly is. MSStats will rely on the information from the other runs and the other features. It's going to assume the features are parallel. So it kind of looks at like, how does this run change? How does this feature change in run 2 between feature 2 and feature run? How do features 1 and 2 change between these 2 different runs? And then we'll try to predict where that value should be based on the different changes, and also including that it should be below this limit of quantification. So that's how MSStats will perform missing value imputation. I did want to mention also that we don't necessarily impute every value that's missing in your data set. So when we do imputation, we want to make sure that we have enough evidence to impute. We don't want to try to impute where we have low evidence, which can really bias the results of the experiment. So some cases where we won't impute, you'll see here, we actually do have some measurements for this MS run 1. So let's say we didn't have any measurements, all of these blue dots were missing. In that case, we wouldn't even try to impute for this protein for this MS run, because we don't really have any evidence of where these values should be for this run. So if you do run for your MSStats and you're wondering, I did imputation, but why are there still NAs in my data set? That's why. And we don't want to bias the experiment. And some issues is like our data can become really homogeneous if we just impute everywhere.
So Devin, would that protein be completely removed from the matrix, or you would still use it? Because in some cases it might be missing because of the biology, if it's like a full-on experiment or something along those
Yeah,
lines.
yeah. So it's kind of a multi-part. So first, if let's say we have like these are both from the same condition, we can still do the condition comparison with just one replicate. Now, if all of the runs are missing for the condition, we can't do differential analysis on that protein, but we'll still return the value in the differential comparison and just tell you like, here's a protein that we didn't have any measurements in the other condition for. You might want to look into it more.
Sure. Right.
Yeah, so you'll still get the protein. It won't just be like disappeared into the code.
And you can see that in another condition, it's high value. Okay. Right.
Yeah, exactly. And sometimes we'll get questions of like, well, maybe like that condition was just all of the values were below the limit of detection. So, that beyond like maybe the protein just wasn't there, maybe it was just like really low. It's just hard for us to determine where it is without any evidence.
Yeah, thanks.
Yep, cool. Okay, so after we do imputation, the next step is feature level summarization to the protein level. We do this with a Tukey median polish. It's a more robust method compared to taking just the mean of the features. So it's really more robust to outliers and noisy data, which you'll see a lot with these experiments. If like one feature is really high for one run, like the mean would just would be very sensitive to that and have that whole value be higher. Tukey's median polish is much more robust, so you'll generally get better results using it. But what we end up having is a single value per protein per run. And this is what we use for the input into our model. So now we've dealt with this subplot, we've done our summarization, and now we can actually do our modeling, and we bring back the whole plot where we have our condition, our subject, and our run with one value per run. And the final model that we're going to fit to this data structure is as follows. So we have just our condition and our subject, and then the technical variation are these runs. So something that might not be initially clear is how do we do differential analysis based on this linear model? So like we have our conditions, we've done all of this work. Where do we go from here? How do we actually do our comparisons? And the answer is using contrast. So if you used MSStats before, you should be familiar with specifying a contrast because we require you to specify a contrast in order to do the comparisons. The exact definition of a contrast is a linear combination of variables whose coefficients add up to 0. And I can give you kind of some heuristic or some intuition on how this is. I think with an example, it's a little bit easier to go through. So let's assume in this experiment, we have 4 conditions, condition 1, 2, 3, and 4. And we want to compare conditions 1 and 2. So we specify our contrast as a vector of ones and zeros, where condition 1 is 1, condition 2 corresponds to -1, and then the rest of the conditions correspond to 0. And obviously this vector summed together would add to 0. And using this vector, we just multiply it by our condition parameters. So condition 1 would be multiplied by 1, condition 2 would be multiplied by -1, and then 3 and 4 would 0 out. And we would just take condition 1, subtract condition 2, and that's our estimate for the fold change. And these condition coefficients are these coefficients we measured in the model here. So we literally will have 4 measurements here, and we just take them and multiply them by a vector of ones and zeros, and then we can do our comparison, and that's our fold change. Now, what's nice about contrasts is they allow us to do more complex comparisons than just comparing condition 1 and 2. So we can really do any combination of conditions as long as the sum adds up to 0. And this is really helpful if we wanted to do averages. So if you remember in that one of those earlier experimental designs I talked about, there were the disease subjects that were measured at a whole bunch of different time points. We could take the average of all of those time points and compare them to just the healthy. And you would do this by just specifying those that you wanted to average out as fractions. So in our example before, we could compare the average of condition 1 and 2 to the average of condition 3 and 4, which is very helpful for complex comparisons. And I'm not going to talk about it too much here, but if you do want to add covariates to your model, say you wanted to add in age or something along those lines, we don't necessarily have a way to just put age into the model, but what you can do is put age into your condition and then average out over the ages, which would correct for the age covariate. So contrasts are really powerful and they allow us to do a lot of interesting comparisons. What would we do with the contrasts after that? I mean, it's really just basic statistics. Once we have our comparison, we take so this L is our fold change, right? We take our fold change, we divide it by the standard error of our fold change to get our t statistic, and then we compare that t statistic to a student distribution with some degrees of freedom depending on our observations. And that's where we get our p-values and our fold changes. So those are the main methods in label-free MS stats. So we have our converters, we do our data processing, including normalization, imputation, summarization, and then we do modeling. So it's a 3-step process, and that's used across all of our MS stats packages. Before we get into the hands-on session, I just wanted to quickly mention some results on between tool reproducibility, since we talked about it a good amount earlier, where we have these different experiments, which are processed with different tools, and we want to have the same differential results independent of what tool we used. So here you can see a couple DIA experiments, which were processed with MS Stats and also with a simpler method, just using our log sum and then a student t-test. What you can see here is we want as many of these differential proteins to be in the middle as possible. We want them all to be shown by the same tools. And if you just compare MS Stats to the other method, MS Stats shows more differential proteins in the middle, and it also has less false positives as well, independent of what tool was used. So 150 to 133, 436 to 413. And that's really our goal when we do we use these different tools and use our different converters. Okay, so I want to talk about just quickly the implementation of MS Stats Shiny, and then we can get into the case study. So the goal with MS Stats Shiny, and here you can see the home page for it, was to take all of this code that we had and try to make it more accessible for people who don't want to work in R directly and would rather use a point and click environment. All of the code is open source, it's available on GitHub, and it's also available as a package on Bioconductor to make it easily installable. And I'll also mention there's a limited version of the app online, but if you're going to do big analyses or large experiments, I would definitely recommend installing it locally and using it on your local machine. So the workflow is the same that we just talked about, right? We have converters, our tool-specific pre-processing. We have our summarization, including normalization and missing value imputation, differential analysis, and sample size calculation, and then we can output the results in a whole bunch of different formats. What I think is really nice about MS Stats Shiny is, we talked about all these, like that big graph of all of our different packages earlier, which is like PTM, TMT, MS Stats, and it's kind of like which do I need to use for my specific experiment? What MS Stats Shiny does is combine all of these different tools into one place. So you'll see here, you just have to select your acquisition type and DDA, DIA, TMT. Maybe we target PTM experiments. And then we have our different converters, which will just change depending on what acquisition you selected. So you don't really need to think about what package you want to use, go through all these different packages, and then choose the right one. You can just go to the MS Stats Shiny and it'll do all of that for you. Another really cool thing that MS Stats Shiny will do, you'll see here one of the outputs is R. And what MS Stats Shiny can do is track your choices in the application. And at the end, once you're done with your analysis, we will provide you the code with all of your choices that you selected in the code version of MS Stats. So this is really helpful for data reproducibility. If you want to know, like, maybe I want to reproduce my experiment later. How do I do that? You can have the code right there. You don't need to go back and look at the log file or something. What's also nice is this allows us to be a little bit more scalable with our analyses. So the MSStats Shiny has a bit of overhead in terms of like RAM it needs and computational resources because it is a Shiny based GUI. And the code version of MSStats doesn't take quite as much resources that Shiny does. So what you could do is run a small subset of your data through MSStats Shiny, grab the R code, and then use the R code to run your full data set. And this also allows us to do some more advanced things. So there's a new package of MSStats that's in development called MSStats Big, which lets you work with larger than memory data sets. And if we wanted to do more advanced things like work with data that's too big to fit into memory, maybe you have like a really big Spectronauts DIA experiment, this would be kind of a workflow that you could use to do that. Okay, so that's it for the overview of MSStats. And now we'll go into the hands-on session. The experiment that we're going to use is a work by Navarro in 2016, published in Nature Biotech. It consisted of 2 hybrid samples, A and B, tryptic digests of human, yeast, and E. coli. And these were at different, mixed in at different ratios. The humans are 1:1, so not differential, 2:1 for yeast, 1:4 for E. coli. There were 3 technical replicates. It was acquired with DIA, and you'll see it was processed with a couple different converters. Are a couple different spectral processing tools. We're going to use Skyline for this specific case study. Okay. So this is my RStudio environment. All we need to do after we've installed the package, which hopefully you were able to look at the Word document or the Google document we sent out, which had specific instructions on how you install and where you can find the data sets. At this point, all we need to do is take our package MSStats Shiny and launch the application. So it's just 1 line of code. And you'll see that the application pops up there. What I would recommend doing if you're running this through RStudio is opening it in the browser. The browser has a couple advantages to using this kind of pop-up RStudio thing. If you're just running it through R, like an R console, it'll just automatically open in your browser. So let's do that. Okay. So now you'll see it just opened in my Google browser. And it gives us a couple things where we can see like iframes and things like that. But this is the homepage for MSStats Shiny. We have some information. So first of all, we'll highlight what version of each package we actually are using. So all of these packages are in active development. We're adding converters, we're refining the methods, everything like that. So it's important to know what methods you're using if you want to come back and use the experiment. And I don't know if you noticed, but we do give this to you a couple times in the code version as well. So you can actually click on these 2 and it'll take you to the bioconductor version of the packages if you wanted to use them. So yeah. Okay. We also have different ways to start the workflow. We have a link to our GitHub, so you can look at the code if you would like to actually go here, and it has some installation things and processing instructions, and then some general notes. What I wanted to bring your attention to first is kind of the help tab, because it's going to give you a lot of information. It's kind of the first place you should go when you're stuck. So a couple different things that in our help tab. First is our documentation. So this really goes into what each tab does and gives more in-depth descriptions of all of the options that you have for label-free, TMT, statistical modeling. And we also are linking here to our user manual. The user manual is, I would say, the best resource for users of MSStats. It's more suited for the code version, but it also does things like give you ideas of how to do your annotation files. It'll show you different ways of, if we wanted to go here, we can look at how we actually need to prepare our data, really in-depth descriptions of the different options. And it's really the best resource for MSStats, in my opinion. Other than that shiny help tab, we also link to different vignettes for MS Stats, which can be helpful for, again, more in-depth description of all of your different options. Okay. Now, if we're ready to run our analysis, all we need to do is click the Run MS Stats Pipeline button. And this will take us to our converter step where we're going to be uploading our data sets and choosing the different types of designs. You'll see I actually linked to the user guide here because I think it's very helpful. We also have links to the PTM documentation and just some other information. We also try to be as helpful for all of these different options as possible by providing different like pop-ups that you can look at. Because especially once we get to the summarization step, there's a lot of different options. So for this experiment, we know it's a DIA experiment that's processed with Skyline. And what I'll do here, we need 2 reports from Skyline. We need our just MS Stats report, which Skyline would actually output the format that we need from Skyline, or Skyline will output the format that we need for MS Stats and an annotation file. So before I upload those, let's just take a quick look at them in Excel. Okay. So this is the output of Skyline provides for MS Stats. So we have our different protein name, peptide sequence, different feature information, the condition, A and B, which we saw before, the replicate, the run name, intensity or area, and then some other things like Q value, which we can filter off of. And this is very specific to Skyline. All of these different converters will have different ways of outputting it, and we have specific converters to handle all the different outputs. But this is the format you will need for Skyline in particular. We also need an annotation file. And the annotation file can be really simple. It gets more complicated for things like TMT, as we'll see in a little bit. But for the annotation file for a label-free experiment, all we need is the run and how it maps to the condition and the bioreplicate. And this is kind of important because this is how we'll tell if an experiment is, say, a time series design or a paired design. So let's say for condition A and B, we actually measured the same bioreplicates, right? Like we can measure bioreplicate 1 in this specific condition, and then later for a different condition. In that case, we'd want to repeat our bioreplicates so we can tell MS Stats that we actually use the same bioreplicates, just different measurements. And like I mentioned in the MS Stats paper, we really go in depth on this because it's really important for how you specify your experimental design. But for now, we'll just keep it like this because we had different replicates. Okay. So let's upload our data. We have our DIA Skyline input file. This is a comma separated file because it's a CSV, and we'll upload our annotation file. Okay. There's a couple other options here that I'll just go over briefly. We can remove proteins with one feature. If we only have one observation for the protein across all of our runs, we can just remove it because we don't have enough evidence. By default, it's unselected. The other option is to have a certain Q value filter, which we could just specify some cutoff for the Q value output by Skyline and filter it based off of that. The last option here is our protein or the analysis level. So I'll go through that in a second. Let's just really quickly look at what happens if I do this with the protein level. So here we have
with the Q value filter, would it filter all the rows for a particular peptide, or only where that peptide, because in the different conditions you might have different Q values?
Yeah, so basically all we'll do is take this detection Q value column and remove anything that's below a certain value. So maybe we will create more missing values like that if we end up having some that are below and some that are above. We kind of just treat them as unreliable.
Thanks.
Yep. Yeah, and you'll see like with these like specific converter options, these are really specific to the special processing tool. So we don't always have a Q value filter. It depends on the tool. But what we get from here is a summary of our experiment. We have a summary of our experimental design, 6 MS runs, 2 conditions, those 2 bioreplicates which we saw, bioreplicate 1, bioreplicate 2. We have a summary of our data set. Now, I really reduced this data set. If we go and process the full data set, it's going to be much bigger, take forever to run. So just for our case study here, we only have 95 proteins, 518 peptides, 3,186 features, and then we can see kind of the intensity range of things. And also the top 6 rows of our data. So if you remember earlier, I showed that MSStats input format or like the converter output, and this will match exactly with that. Going back to this analysis level. So this is a feature that was requested a little while ago for MSStats Shiny. Some users were wondering if they could do peptide level analysis with MSStats. So instead of looking for differential proteins, look for differential peptides. We can do that. So if we wanted to click on the peptide button, we can upload our data again, and now we'll see in this protein name column, we just have our peptide. So this is definitely possible, and it's something that you can do. I just want to say that our methods in MSStats are designed for protein level inference. So things like missing value imputation, summarization, they work better with more features. And if we do peptide level analysis, we'll have less features per peptide compared to the features per protein, and the analysis methods may not be as stable. I can't confirm based on the experiment or based on any experiment what it'll look like, but I just want to make you aware of that. But it's an option if you do want to go down that road. Once we're satisfied that our data was uploaded correctly, it looks good here, we can move on to the next step of the analysis. And this is data summarization. So this has all the options we talked about before: normalization, imputation, summarization, with a couple additions that are kind of a little bit more minor. So first off, we can select how we want to transform our data. It's going to be a logarithmic transformation, but we can change the scale, log 2 or log 10. I'll just keep it log 2 for now to make it pretty simple. We talked about normalization, and here are our options. So equalize medians is the default. We can do no normalization. We can do quantile normalization, or we can use global reference standards. The feature subset, this is really helpful for experiments like DIA experiments, where you might have a whole bunch of features per protein. Like, I mean, it can get into like the hundreds to thousands of features per protein. What we've seen at some point, using all the features is redundant and will just make your processing time take a really long time. So what you can do is use either the top features, and we define top features by the maximum sum of the intensities, or we have this remove uninformative features and outliers, which I'll talk about in a second. What I'll do now, just so that we can look at the information, is just process it with all features, and then we can reduce the number of features and see what changes. We have missing value imputation, which is like kind of what value you think, or what value the tool specified as missing. So some tools specify all values as random, either between 0 and 1. This is going to depend on your specific tool. I'm just going to leave this as the default. We have our quantile for censoring, like we talked about. We look at that histogram and to try to decide where the values are unreliable. So we can reduce that quantile, and maybe we think anything below like the 99th quantile is unreliable. And lastly, we can choose to do imputation or not. So now I'm just going to run summarization so we can look at the output. So the first thing we have here is our quantification table. And basically, we just look at the summarized value. We can look at it based on the biological replicates or on the condition level. For this, because we only have one biological replicate per condition, it's not going to be that interesting if we change it. It still just shows A and B, one column. But what you can kind of do is look, okay, well, for this protein, what were these final summarized values for that protein? What's going to be our input into the model? And we have 6.379 versus 6.559, which is good because we expect this human protein to not have any change between conditions. But maybe for like an E. coli protein, which we do expect to change, you can see there's a pretty significant difference, 4 versus 6, which is nice to see. So this is a good way to look at kind of our summarized values in really a compressed format. We can also download this data. So let's just do it in long format because it's the format I prefer. Can download the data. And then we can look at it in Excel. So here's the data in Excel. Same thing. We just have it now in like a bigger format that we can go down and look at everything. Okay. Next option here is the summarization plots. So we include two plots here for you, quality control plots, which are going to be the box plots and profile plots. Let's look at the quality control plots. The first option is for all proteins. And these are these box plots that we were looking at before for normalization. You can see here that the medians across all of these were normalized, right? That's what we selected. And it's kind of hard to tell because of the scale and also because of this experiment in particular. But in other experiments, you might see that these interquartile ranges are pretty different too. All that matters for the equalized medians is that these lines are equal. If we wanted to do quantile normalization, we could do that as well. And you're not going to see much change because it's already pretty close to being equal, but now these are actually equalized based on these boxes. So let's go back to equalizing the medians. Let's save this plot, which will allow us to see it here. And hopefully I can show some differences if we don't normalize. So now I turned normalization off and we will look at the box plot. We'll save the plot, open it up, and let's compare it to what we had before. Here it is. Okay, so this is without normalization. This is with normalization. And it's still kind of hard to tell just because of this specific experiment, but now these normalization lines aren't exactly equal. So there's a little bit of differences, maybe like here and here, you can kind of tell differences because we didn't perform normalization. And we're going to be able to see that as on the whole of these all proteins quality control plot. We can also look at the individual proteins. So you can look at like the humans, which we expect to be pretty similar, or maybe E. coli, which now we expect a pretty big difference, which we can actually see. The last option for normalization is if we have global standards. And we're not going to do this because we don't have global standards, but I just want to show you how it works. We can choose our standards if they're proteins or peptides. And what it'll do is we'll click on this button and we can choose what our standards are. So what if we wanted our standards to be the human or E. coli or something like that? We choose which ones their standards are and we process normalizing to those. But I'm not going to do that because we don't have standards. So let's go back to our equalizing medians and let's look at the profile plots now. So let's look at the human profile plot. And we can immediately see, because this is a DIA experiment where we have a whole bunch of features, the legend is really big. So let's remove the legend so we can get a better look at it. And we still have a whole bunch of features, right? It's kind of hard to tell because they're overriding each other. Like what's actually happening? Is this protein changing between A and B? What's nice about the profile plots is we can look at a summary. And now we can see with the summary, this feature is fairly flat. It's even though we have a whole bunch of features, some of which have some noise, in total, the feature just looks flat. Now, what if we wanted to look at something with E. coli, maybe? Okay, well, E. coli, a lot more noise, but feature A is much lower than feature B. And we should see the same thing with yeast, but to a lesser extent, since we had a 2-to-1 ratio compared to 4-to-1 with E. coli. And now we can see this yeast one is very low. Well, you can tell with this yeast one is in particular here, we have a whole bunch of features. And I will save that. So we can see we have both of the feature level plot and the summarized level plot. So maybe we don't want to use all of the features in our analysis. So let's just try to use maybe like the top 10 features for this analysis, where we'll just take the top features with the highest intensities and look at the results there. And now you can see there's a pretty big shift in the summarized value. So we by default will show still all of these features, but you can see now that we only use those really high intensity features, this profile plot really shifted up compared to what we saw before. The other option here is these removing the uninformative features and outliers. So we can see a lot of features here which have a lot of variability. So we have some that drop really low, then they jump really high again. These ones are really high, then they drop low, then they go up high. And these are really noisy features. Maybe they just had some artifacts in them that caused them to be really noisy, have outliers, have issues. We have probabilistic methods built into MSStats to detect this for you. So you don't need to decide which of these and do it are messy and do it manually. So I'll select remove uninformative features and outliers. And this will take a little bit longer to run because we have more methods behind it that need to process. But now you can see the features that we end up with are a lot cleaner. So we don't have those ones that are bouncing around. We don't have the really high values or the really low values. We have features that are much cleaner, and our final result is just these high quality features. So what I would recommend you do is play around with this on your own experiments, kind of look at the profile plots. If you use all features, maybe if it's a really big experiment, you're like, this is taking forever, just kind of reduce the number of features. If you think the features are looking really messy, try to play around with this button for removing the uninformative features and outliers, see what the results of the differential analysis are, and kind of look back and forth empirically and try to kind of decide what's best for your specific experiment. What's nice is we provide you a lot of options in order to do this. So you have a lot of ways to go about the data processing. And like I mentioned earlier, these are really important, especially normalization and the feature selection and imputation are going to be very important for what actually comes out of the differential analysis. So the final option here is downloading the data. And we provide two different levels of data for you to download. The first is feature level data. And this is going to be the pre-summarization data. That's before you actually get like one value per protein, but it's going to be log transformed, normalized, and with missing value imputations. So it'll be a bigger data set or a bigger file here. We have all of these specific feature level information, protein, the peptide, the feature, which is our transition plus the peptide. And then we have like our untransformed intensity, our abundance, and our predicted value for imputation, if we did imputation. The other option here is our protein level data. And the protein level data is the summarized data. So now we have no features, no peptides, but we have one value per protein per run. And we have it transformed on the log scale for this run, this group, this subject. We can see how many values we're missing. So it's kind of like a quality of the feature, like, or of this run where there are a lot of missing values. How much do we need to impute? We're more than 50% missing, all of that kind of thing. So you can do some diagnostics on that level. Okay. And the next step of the platform is to do statistical modeling. So first we need to specify our contrast matrix. Now this one's really easy because we only have two comparisons, A and B. There's only one thing we can do, but in our TMT experiment, which I'll show in a bit, we'll have more options and we can do more advanced things. So this will start mattering more, but for now, we'll just do something really simple and we'll run our group comparison. And what will pop up here is differentially abundant proteins. So we can see E. coli, which is what we expect. You can actually see at the top a human one, which is a false positive. We know human was actually not differential. Luckily, there's not too many humans here. Most of them are E. coli, yeast. There's a couple humans popping up. And we can download all of our modeling results, including ones that were not differential or just these differential proteins. And we can see this is our classic data table for the full modeling results where we have the protein, the comparison, fold change, standard error, t-value, degrees of freedom, p-value, and adjusted p-value. And going back to Soyed's question, this is actually where we would show if we didn't have any comparisons for, or any measurements for one of the conditions, it would be in the issue here. It would say completely missing. I don't think we have any here. No, it would be completely missing in one protein or in one comparison, but we would show that here with just an infinite fold change and a 0 adjusted p-value.
Cool. Perfect. Thanks, Devin.
Yep. Okay, the other thing we have here is our volcano plots. So we can display one plot in the browser. You'll see these blue ones are our E. coli proteins, which we expect to have a big fold change. We can see our yeast proteins here, which have a small change, but still are differential. And then we see a couple that are really close to 0. These are probably our false positive human proteins, and some that are a little bit higher, but not differential. Maybe these E. coli peptides had a high variance. So while they had a big fold change, the variance was high. So we didn't have a significant p-value here. So you can really quickly do kind of an analysis of this by looking at which ones might be interesting to check out later. Maybe we want to look at these ones or something. The other option is our comparison plot, our heat map. Heat map's not going to work because we only have one comparison. Heat map needs at least 2 comparisons so we can look at the changes between the comparisons. The last option is our comparison plot. And what a comparison plot will do will show us the mean for each condition, or each comparison will show us the mean and the confidence interval around that mean. Thing to take away from a comparison plot: if the confidence interval is over 0, that protein will not be differentially abundant. We'll show it as insignificant. So this is a human one. Let's look at an E. coli one. Now we can see this confidence interval is very far from 0. So we know this one is differentially abundant. Okay, we can also download our analysis code like I talked about. So let's open that up, which might have a problem. Let's see if we can do it through this. Okay, let's see. Downloads. There we go. So this is the analysis we just did. You can see that we have the package versions that were used. We have the converter. You just have to input your CSVs where the file paths to where they are, and we will do all of that for you. We have our data process where we select equalized medians. We used high quality features in the end there. We have examples of how to create data process plots. We create your contrast matrix for you, which I really like because it's kind of a lot of typing to create your contrast matrix in the code. We do the modeling and we do our modeling plots. So that's what the code looks like there. And the final step is the design for future experiments. So we use the variance that we had before. We treat that variance as what we might see in a future experiment. And we can define, maybe we want like a 0.9 power, like we want a really powerful experiment. And we do expect to see maybe a bit higher fold changes. So maybe fold changes that go up to 2. And we can mess around with the false discovery rate. So as the false discovery rate goes up, we need less proteins, or sorry, less biological replicates. If we want like a really low false discovery rate, then we'll need more biological replicates. So you can play around with this and kind of see, like, to detect fold changes of something low like 1.25, we actually need more biological replicates of like 6. So that makes sense too. Okay, so that's MSStats Shiny and how you kind of run through it, some of the different options for label-free DIA. Before I go into TMT, which will be a little bit shorter, are there any questions?
Maybe I can ask one while people are thinking of questions. I keep thinking to your comments right in the beginning about power, and that is not only determined from variability, but also number of features or proteins or peptides you're trying to measure. And the more of those you have, the more replicates you need. So I mean, there's definitely a benefit if you're doing a discovery experiment to filter out all the noisy data before you do any statistical analysis. You actually have a better chance of picking up differentially expressed features or proteins in that way, rather than trying to keep too much of that noisy data and then just drowning out the interesting bits in noise.
Right. Yeah, and I think there's a couple levels to that, right? So you have like, just in general, noisy proteins that maybe you would want to eliminate. Like, proteins that just in general have like a ton of missing values, a ton of summarized value, or a ton of like, outliers and things like that. And if we include those, they're going to be included in our p-value adjustment, right? So just all of these noisy peptides are going to be lowering our adjusted p-values to begin with. Beyond that, we have like noisy features, like we talked about, like outliers and noisy measurements there. And that's also going to have a pretty major impact on the final values. So yeah, I think with these with these experiments in general, there's always like the overall protein level and the feature level, and it's kind of like a trade-off, and you have to look at both of them and try to decide like what you want to do with each section.
Yeah, this makes sense. Thanks.
Yeah.
So we have two questions that come in. I don't know if you can see them in the.
Yeah, I can. So the first one, is it possible to generate labeled volcano plots using MSStats Shiny? So in MSStats Shiny, I don't think it is. Let's double check though. Oh, look, it can. Okay. Yep, so it is. So this will only show the labels for significant proteins, and you can see it does kind of like get a little bit on top of each other. We can obviously save this to a PDF, which might help us a little bit, or if not. So it's a little bit tough if you have a bunch of them, but you definitely can. And I will say with these plots, there's a lot more options in the code version. So we can only have kind of what we build into this UI, but there's probably 20 parameters to change the axes, change the labeling, everything like that in the code. So if you want more flexibility, the code version is definitely the way to go for plotting. Okay, cool. That was the same question. Perfect. Cool.
Okay, so no more questions at this point. Maybe we can carry on and see if something pops up. I'll let you know.
Awesome. Yeah, let's do that. Okay, so let's go back to the slides. And so now we're going to talk about MSStats for TMT. This will be a lot faster because we are building on all of these methods that we've worked on for label free. TMT introduces new challenges that we have to address with these methods, but the general workflow, the underlying models and methods are going to be the same. So let's look at a standard profile plot for one protein in a TMT experiment. So there are more challenges because of the complex design. So this is a profile plot where we have the log intensities on our y-axis, channels on our x-axis. These different runs are made up of, I think there's 5 different mixtures and 3 technical replicates per mixture for this experiment. But what you can see here with these 10 channels per run, we have some conditions here. We've done good randomization over the channels where we're including conditions in each of these mixtures. We have a normalization channel, which I'll talk about soon. We have different sources of variation. So now we have 11 features within a TMT run. We have within peptide variation. So maybe this pink peptide, we can see it kind of goes up and down within its own feature between the conditions. We also have between mixture variation, where we have these red peptides or these green peptides, which change. So like in run 4, it's kind of lower, in run 5 and run 3, it's that green one, it gets higher. And then we have our standard within subject variation. So now we still have like the changes between biological subject 1 and biological subject 2 in the same condition. And so what happens with these TMT experiments is now we just have more sources of variation that we need to deal with. And that's a challenge, and we have to update our models to deal with all of these sources of variation. Another big issue or big complexity that comes with TMT experiments comes in the missing peptides between runs. So let's look at first this this light blue peptide in run 3. Hopefully you can see. You'll notice that it has only 4 values. So it was missing in a bunch of these different channels in this mixture. what's nice about TMT is that this problem is not as big of an issue as it was for label free. So for label free, we might have like 50% of the rows or 50% of the runs missing a feature. Within a Plex, within a mixture, we really will mostly see all of the values for that protein. And that's just due to the labeling of TMT. So you can see like that's really the only one. I can't really see any others here that have major missing values. So they're all, otherwise they have all their values. And that's a really big advantage of TMT. However, there is a second type of missing values, which is actually a really big problem. And that is missing features between plexes. So let's look at that blue one again, this blue feature. We don't have it in run 1 at all, don't have it in run 2, we have it in run 3, have it in 4. It's really missing in most of these runs. We only have it in 4 out of the 15. And that's a big problem because we talked about before, if we don't see this feature, we really can't do imputation because we don't have evidence of like where this feature should be in these other runs. So what ends up happening is these runs become less comparable. And if we can't do imputation, we will have different features that make up our summarized value for each of these mixtures. And this issue only becomes bigger as the number of mixtures goes up. So really, if we have more than 5 MS runs or 5 mixture runs, we can expect to have around 50% of the features being missing. And there are very few features that are across all runs. So I think the only one is this green peptide, although the other one has a lot too. So we have just a ton of missing features across runs, and that's another challenge. So let's go back to that data structure we had for label free, how we see a normal label free experiment. Well, now we have more sources of variation in terms of the actual mixtures. So we need to add more to the model. So this is how we look at a TMT experiment now. We have our different mixtures. We can have different technical replicates from the same mixture sampling. In those technical replicates, we have our conditions, and under our conditions, we have our bioreplicates. And the features look pretty similar, but you'll notice we have more missing values in terms of across MS runs. So, we're totally missing like all of these ones. We have all of the runs here, and we're missing very few in terms of like, if we do see the feature across channels, we see it across most of these channels. So it's very often, very rare that we actually see one missing in terms of like this feature for the whole channel. So that's the experimental design. Let's look at that's how we see the experimental design. Let's look at the workflow again. So it's really the same workflow like I talked about. We just have more complexities. So we do have more things we have to talk about with the experimental design. So we really want to be able to, when we allocate our conditions and our bioreplicates to the mixtures, we want to make sure we have a good combination of conditions and bioreplicates in each mixture. So say, going back to here, we don't want to have one mixture with like 0.15 in all of our channels, right? We want to be able to include all of our conditions into each mixture in separate channels. If we just had 0.15 in all of them, we would have a very biased experiment and the mixtures wouldn't be comparable. So it's really important to think about how we're going to allocate our conditions, replicates to our different mixtures. Beyond that, we really recommend having a reference channel, one channel set aside for reference channel normalization. And I'm going to talk about this more in a couple of slides. This is really important to make our mixtures comparable, make our mixtures comparable across all of the different runs. The last thing we have with with TMT experiments is fractions. So we see fractionation a lot with TMT experiments, and we have different methods to handle fractionation. So here's the workflow. Same exact thing as before. We have converters for a couple of different spectral processing tools. They're going to be a little different, so depending on what actually does TMT. We do our filtering. which is the same as before. I mean, we can do shared peptides, which I don't think I talked about. Shared peptides are where we have a peptide where we can't identify it to a specific protein. So say we have one peptide that could have gone to protein 1 or protein 2. The special processing tool couldn't tell us. Do we want to keep it and try to model it, or do we just want to throw it out? We can also keep, if we have multiple PSMs, we can keep like the best one, which generally means like the highest intensity one. And lastly, I've mentioned fractions. We have ways to combine fractions. We can either take like the mean of the different fractions, or we can take whatever has the max intensity, or we can do the fraction sum. A couple different options, just depending on what you want to do. Okay. So in terms of data processing, it's very similar. So normalization, imputation, summarization. We do have one extra step. So spectrum level normalization, which is the same normalization we talked about before, imputation, if we can do it in TMT, protein summarization, and then this protein level normalization, which is really important. So first is spectrum level normalization. And this is just that equalized medians that we talked about before. So we can just take all of these medians, they're a little bit off center across all of the different mixtures, and then we'll just normalize them all so that the medians are the same across all of the runs and channels. Next, we have summarization, which is done with Tukey's median polish. Same as before, we just summarize the values into one value. What's worth mentioning here is because we have different features that are used for our summarization, these summarized values are not necessarily directly comparable. We can't just use them in our model directly. And this is where the next step comes in, protein level normalization, where we use a reference channel. So this is why we recommend having a reference channel, so we can see how the reference channel changes between our mixtures, and we can use that to normalize our summarized value and then make them comparable, even though they were on different scales before. Now we know why the scales were different. We can update them. And with the reference channel, we get much better results than if we didn't have a reference channel. We've also tested having 2 reference channels. It works well. We did see a slight improvement, but we did kind of notice that it's better to allocate the other channel to more samples. So you don't really see enough of an improvement for it to be worthwhile. So 1 reference channel is what we recommend. Okay. So then we have our modeling and statistical inference. And we do have this really big data structure, so our model is much more complicated. We have this mixture, we have these different compounding effects between the different variances, between our different parameters. So what we do again is we just fit a subplot, right? We do our summarization, and now we have a much simpler linear model. So it's really the same as before. We just have this mixture and then a technical replicate per mixture variables. So we can control for the mixture effect essentially. But once we get this, we get these coefficients for our conditions, and we do a contrast comparison to estimate the comparison. So it's really the same workflow as label free. We just have some more challenges because of the different mixtures. So we have to control for them and also do some extra normalization, different imputation, things like that. And the last thing I want to show for TMT is just a quick comparison that Ting did on the different results. So we have 4 different methods here. We have median normalization with Lemma. We have this EDGE-R method. We have the methods built into Proteome Discoverer, and we have TMT. The first thing you'll notice in terms of the actual testable proteins, EDGE-R reported much less testable proteins. And that's because EDGE-R actually removes any proteins that are missing more than 60% of their values are missing. So in the data set that we tested here, there were a lot of missing values. EDGE-R was like, I can't handle any of these that are more than 60%, so it just threw them out. On the other hand, Lemma did have a lot of testable proteins. We had a lot of true positives for Lemma, but we also had a lot of false positives. So Limo was very sensitive. It reported a lot of false positives and had a high false discovery rate. TMT and Proteome Discoverer were a bit more comparable, where they had similar true positives, and PD had a couple more false positives. So while they're comparable, MSStats TMT still showed the best results with the lowest false discovery rate. And all of that's just to give you some confidence that these methods that we developed with dealing with missing values, dealing with or having our summarization and our models, it does do a very good job of handling all these complexities in TMT and producing the best results possible. Okay, so I kind of just breezed through it, TMT. Again, if you understand, if you know the results or if you know the methods for MS stats, the TMT ones come along really are pretty straightforward. I would think. So let's go into the hands-on session. So we have another data set. This was actually used in our MS stats TMT paper. It was another controlled mixture with 48 UPS1 proteins spiked into some background. They were spiked in at different levels. So we can see these UPS1 proteins are supposed to be differential between comparisons. We generated a reference channel by pooling all of the UPS1 peptides, and we profiled it in 3 MS runs. So 15 runs from 5 mixtures, and it was processed with PD. And before I go into so I'm not going to come back to these slides. Before I go into the case study, I just wanted to thank a bunch of our collaborators. So our lab is a purely computational lab. We don't do any experimentation directly. We really rely on our collaborators, and obviously we're integrated with a bunch of tools, so it's important to collaborate with the different tools as well. So our collaborators are very important. So everyone in Olga's lab at Northeastern who's helped develop the methods, Mateus, who's done a lot of method development, our collaborators at ETH Zurich, which really helped with developing MSStats LIP, collaborators at University of Washington and Michigan, which helped us build our converters and integrate their tools, FragPipe and Skyline, directly with MSStats. Arop, who worked on MSStats QC, collaborators at Genentech, including Mina, who's now Genentech, who are very important. They really help us develop a lot of our methods, including MSStats PTM. Collaborators at UCSD, who have worked with Massive Quant, and our collaborators at Pfizer, who helped us benchmark our PTM methods and also integrate some work with MSStats Shiny. Okay, so let's do the case study with MSStats TMT. So I have the same thing that the same platform that we have opened up here. What I do for when I want to process new experiments is reset the pipeline. What that'll do is just clear out everything, all of the variables that we had saved, and just make sure that we don't run into any weird bugs where like it had some old variable saved that like is now trying to go into a new experiment. Just makes things a little bit easier. Okay, so same way. Now we have a TMT experiment that's processed with Proteome Discoverer. We can still select the analysis level protein versus peptide, which we'll skip for now. PD, we have two files still, but they're going to be a little bit different because now we are on a different tool. So this is the PD input. It's wider, it's in text format. We don't need to look at this too much, but what I do want to open up is the annotation file. So this is our annotation file. You'll immediately notice that it's a much more complex than what we had previously. And this is because we have a lot more runs and we have a lot more channels and more mixtures. So we have our runs with a specific name, fraction, the technical replicate per mixture, the channel name, the condition name per channel, the mixture that it is. So you'll see we have 3 technical replicates which were run using the same mixture, and then our bioreplicate. And we can still do time series designs. We can do pair designs in TMT. So this can get really complex.
Devin, we have a question about the Shiny convert PD data for MSStats analysis.
Yeah, definitely. So that's what I'll show right now. So we have our PD analysis, right? So it's the same thing as before. We just upload our text file. Oops, that's DIA. That's not what we want to do. TMT, our PD input. You'll notice this is a text file, so it's important that we select the right separator and the annotation file. So yeah, basically anything you can see here, if it's highlighted, we have a converter for it. So FragPipe, SpectrumMine, OpenMS, PD, MaxQuant, for TMT. It depends on these, so. Okay, so let's upload the data. And here it is. So we have, a more complex experimental design. So now we have more mixtures. We have our technical replicates, our runs, conditions. Again, I took a subset of this data because this is a pretty big data set. So we only have 99 proteins. You'll see we have less features. I think last time we had like 3,000 for the DIA data set. And we also have a different input, right? So now we have our mixtures, we have our technical replicate per mixture, our channels, but it's a very similar format. I mean, we just have a couple more sources of variation. And you'll see here, we don't actually have a Q value filter here for this. One thing with MSS Shiny, because we have to build in everything manually into the platform, there might be a couple more options for like the PD converter or some of these converters that are in the code. You can kind of look at that yourself and see like if maybe you need those filters or not, depending on your experiment. Okay, so let's go to the data processing. You'll notice here we have less options. We don't give you the option in MSS TMT just has a couple of different options. It still uses MS stats in the back end, but we don't give you all of the options that are in MS stats. So like you have to just do log2 transformation. But we have our peptide level normalization, which is that first step where we're doing just our global median normalization. We have our summarization method, and we give you a couple extra options here. You don't just have to do MS stats, which includes TMP. And some other more complex filtering. You have some more simple options as well. We have that cutoff, again, that censoring cutoff, local protein normalization, which is that reference channel normalization, and filtering. So we can remove the normalization channel, or if you want to keep it so you can look at it. So let's run this. Takes a little bit longer because we need to do every run separately now. So it's like you're running the MS stats analysis 15 times. But the results the actual results we see in the platform are pretty similar, right? We have our summarized results and we have a couple more because we have a couple, actually said the same thing as before, where we only have one actual biological replicate per condition, but you can see the protein. These are our spiked in proteins that we expect to be differential. So you can kind of see they do change across these different conditions. These are our background proteins. They should be the same, which they look pretty much they are. Can download it. Let's look at the summarization plots for TMT because I think they're pretty interesting and there's a lot more that goes into it. So here's our quality control plot. Kind of hard to tell because there are a lot of different mixtures now, right? So maybe for this, you would want to save it so you can kind of look at it more in depth. Just because like of how many there are and it's really complicated. And I think it'd be tough to tell, but without selecting this, this is how we equalize all of our medians across here. If we didn't select it, then our medians would be a little bit different, but they aren't. So we'll just keep them like that. We can look at individual proteins as well. So let's look at this one, which we expect to be pretty flat. You can actually tell like for this run, we had no features, right? So we won't even be able to use that in our statistical analysis. Some of them only had like a couple features. Look at the profile plot for this too. Okay, let's remove the feature legend. And we can immediately tell kind of what we were looking at before, right? Like we have this orange feature is missing in some of these runs. This green feature is missing in a couple of runs. We only have this blue feature in 3 runs. So without the reference channel, these would be very hard to compare. So let's look at the summary. Okay, we can see that they're all equalized, right? That summary is exactly the same because of our reference channel. But if we didn't do reference channel normalization, let's see what it looks like. Okay, So now you'll see that without reference channel normalization, these summarized values are really all over the place. And that's why we really recommend having a reference channel. It's really important so that we can make these summarized values, regardless of the mixture that they're used, regardless of the missing values, we can actually compare them and we can ensure that your modeling results are going to be of high quality. So definitely reference level normalization is recommended. Or reference channel normalization, sorry. But let's go back to reference channel normalization for our modeling so that we get the correct results here.
Do you see a similar trend if you do DIA, a global standard normalization? You maybe get a little bit better normalization versus medium, or?
Yeah, well, I definitely think if you do the global standard normalization, that's the best way you can go about. Because then we're really seeing the true technical variation that we can remove it. So if you can do it, I would definitely recommend it. It's probably not as big of an issue as TMT, just because like with the features we see in DIA, they're probably going to be the same across runs. So it's not quite as big of a deal, but definitely that's the way to go if you can do it.
And do you have a feel? I saw that you can select more than one double standards. Do you have a feel for how many peptides or proteins you want to have the double standards, or one would be sufficient?
I would say probably as many as you think you have a good handle on. So, if you have them spiked in, obviously you would just pick the ones that you spiked in, right? All of them.
Yeah.
If you expect, if you're like expecting some proteins not to change and you want to use those as your standards, I would pick as many as you can because
Gotcha.
maybe there was a couple that had like some technical artifacts or something like that, or maybe, I don't know, maybe there's just like there's some issue in a couple of them, and selecting more would give you a little bit more robust, yeah, analysis.
Okay, thanks.
Cool. The last one, I just want to show UPS proteins so we can kind of look at it. These ones should change, right? And we can kind of see like while they're on the same scale, they do have different, within the mix, they have different levels. We already showed the downloaded data. It's the same thing. We have our featured level data. We have our protein level data. Now we have our contrast matrix, and this is going to be a little bit more complex. So I selected a full pairwise comparison, which will compare all of the conditions to each other. But we could do things more complicated, right? We could compare all against one. We could create a custom pairwise matrix where we only want to compare some of the comparisons. Or we can do a custom non-pairwise comparison. And this is where I was showing you those fractions, those changes in like maybe we want to do 1/2 and 1/2 versus -1/2 and 1/2 or something like that. So you can do all of this in MSStats Shiny directly. And like I mentioned, I really like it because then you don't have to like code in all of these like vectors and matrices and make sure you're adding the names and making it all work. And it's just a lot easier, in my opinion. The last thing I wanted to talk about is empirical Bayes moderation. So a lot of times we get the request is like, why are your methods like, what's better between MSStats and Lima? And what Lima does is it assumes that all proteins in your experiment have the same variance, which is a strong assumption. So if you assume that all proteins in your experiment have the same variance, if you have some proteins that have a really high variance or a really low variance, you can moderate those towards the median. And what that'll do is show you, generally make your experiments more sensitive because we're moderating down all of these really high variance proteins. We can do that for MSStats TMT. It's not implemented for MSStats label free or base MSStats, but we do have it for TMT. It's a strong assumption, right? Like you're really saying like all of my proteins have the same variance, which might not be true biologically. So this is something I would just recommend kind of thinking about before you actually go into it, because it's a pretty big option and it'll definitely change our statistical analysis results. Okay. So now we have our results. Same thing as before, we would expect to see the UPS proteins here. We would expect not to see the these ones here. You'll kind of tell like these ones that are false positives have very low fold changes, but they also have very low, standard errors. So the final result is having, low adjusted p-values. We can look at our volcano plots. So we'll look at one here. One thing to mention is that if you wanted to look at all of them, you'll get an error if you want to view them in the browser. We just have to save them to a plot, and then we can look at all of them at once. We can also look at our heat maps, and now they should actually work, where now we can see the different comparisons. We can see our different proteins. These are our true positive proteins. These are our false positives. And you can see across most of our comparisons, our true positives are downregulated. And we have our comparison map here, which again, it's the same thing, but we have more comparisons. So if you'll see, like, these are really tight standard errors, really tight confidence intervals. So if they're over 0, they'll be insignificant. If they're not over 0, they'll be significant. And we can look at maybe the UPS one, which should be, yeah. So they're all far from 0 because these are our true positives. Other than that, I mean, all the everything else is basically the same. We can look at our code for TMT, which will have different functions. Oops, let's do that real quick. Okay, so this is our TMT code. Now we're grabbing a different converter. Right now we have the PD to MStats TMT converter. We're using MStats TMT functions, MStats TMT plotting. We created that contrast matrix. It took a good amount of code to do that. And we use our TMT group comparison. But like I said, with the MStats Shiny, I mean, it's all in the back end, right? It kind of looked really similar to base MStats. You don't even need to worry about like what function you're using and things like that. So yeah, that's MStats Shiny for TMT. Do we have any questions on TMT? I kind of went a little quicker through it, so hopefully if you have any questions.
Maybe I'll ask a question. Maybe it's not so much on TMT, but these comparisons, are they available also if you're doing like a label-free DI experiment? Maybe you've got like a time course experiment or something on those lines, and you can
Yeah,
like these features.
like which like this comparison plot or like these kind of things, or
Yeah, yes.
yeah, definitely. All of the plots are the same. I just couldn't show it for the last experiment because we only had 2 conditions.
Yeah.
But yeah, definitely all of them are the same. We actually use the same function to plot them, so definitely.
Fantastic.
Cool.
So guys, if you don't have any questions, we'll close up the session. I think I have to say that it was super informative for me, hopefully for everybody else. We have recorded it, so I'm going to put it up on our YouTube channel. So check out Resin Bio YouTube channel, and then it's going to be recorded, so you can go back and rewatch it. I'll definitely be doing that. And it was, yeah, very informative. I want to just thank you very much, Devin, for this. Yeah, see you at SMS, I suppose, and hopefully some of you at SMS next week. Yeah, thank you very much.
Awesome. Thanks for having me, Stein.
Well, thanks for coming along. Cheers. Bye-bye.